So this lecture relates to the genetic code. So you see, the first part will deal with what is essentially called the genetic code, how it is translated, and on the second part I will address something which is booming now in the field, and this is related to the title of the talk, and how can we expand the genetic code, that is to use the tools, the biological tools which are available to produce extra amino acids bound and incorporate in the proteins. So it's a biotechnological approach. It's not really, although there are links to the evolutionary aspect, but it's a very useful application. And it is not a surprise that Jason Chin, who was a postdoc with Peter Schultz, who is a pioneer. Peter Schultz in California was a pioneer in these trials to include extra amino acid, other amino acid, into proteins. And Jason Chin, who now has his own group at the MRC in Cambridge, changed the techniques and made it more biological. And he got the EMBO medal for young investigator last year for that work. So I thought it would be interesting for you to see what can be done and to use these tools for applications. There are a lot of useful applications now in research. So we'll come to that. That's the overview. But let's first go to what is the genetic code and the translation of genetic code as it is, and the questions why do we have such a rigid and strictly controlled genetic code? And so for today's translation apparatus, and so we are a bit far away from the talk we had on Monday, where we were talking about the evolution and going to the origin of the genetic code. We'll speak a little bit about that, but here today we are not going to do that. We are going to analyze today's genetic code, which is the end of a long process of evolution, and the key players in that process for the translations are the amino acid tRNA synthetase, so now they are proteins which are doing the work, and of course the other key player is transfer RNAs. So transfer RNAs, you have seen what they do once they are charged on the ribosome, and they are doing lots of other things. So tRNAs are very key molecules. So the main function of amino acid tRNA synthetase, because there are other functions, these are very complex molecule modular, is the deciphering of the genetic code. And already 20 years ago, it was found that amino acid tRNA synthetase, which is the family of enzymes which translate the genetic code, was indeed not one family, but was one family, but there were two classes, not just one, as everybody thought before, because you had one reaction charging the transfer RNA, and then so it came as a surprise when we found that, in fact, there were two classes. And the two classes here, you have the partition, and what came clearly at the beginning of the discovery, when this partition was found, is that the partition was based on structural consideration, chemical considerations. Why did we have two enzymes, two classes of enzymes with totally different enzymatic core centers for the same reaction, which is attaching the amino acid to the transfer RNA on the CCA end? So that was not expected, so different. And the solution of that was that, fortunately, before this structural work was done, people were investigating several aspects of this, of transfer RNA life. And for example, one of the questions which was raised, you know, the terminal CCA and the terminal ribose has two hydroxyl groups available. The 3' because the chain is stopped there, and the 2' because it's an RNA. So there are two hydroxyl groups. So the question was where does the amino acid be attached? And in fact, it turned out that the partition was linked to that key operation. These are the 2' hydroxyl. I mean, these enzymes are attaching the amino acid to the 2' hydroxyl and this class to the 3'. Because the chemistry was available. People didn't know the answers. You know, that was the Göttingen group from Kramer, and then also Rich at the MIT who did that work. That was a blind search, so you know, chemical probes, and so we had this partition available. But you know, when you have the genetic code with 2', 3', you know, it was a bit difficult to make a correlation. When we had that, we could see, oh, that's the correlation. Then it makes sense. Once you have things like that, where you have a functional differential, you say, well, what is so different between these 2' and the 3'? And what is different, here you have the explanation. Here you have the adenylate, the 2' is going on this side, the 3' is pointing on this side. So you have, with the 2' and the 3' hydroxyl, you have two different chiral centers. And chirality is an essential part of life of structures. When you have a chiral center, an enzyme, if an enzyme charges one chiral center, recognize, it cannot recognize this enantiomer, the different chirality. That's essential. And so in chemistry, you will never, just by playing around with few changes, make a 2' charging, a 3' charging. Actually, people have been tried. It's a totally different recognition process. That's a basic division in chemistry. So that was the explanation. And you will see, so these are the data. When you have these, you see, every rule has its own exception or fuzzy thing. You see that they all obey that, except here the cysteine, where chemically it was difficult to ascertain which was the one. The tyrosine is also an ambiguous one, and the phenylalanine. Phenylalanine is the only real exception. Belongs to class 2, but charged that. So today we are not going to go to details for that. But then as a consequence, also the main division of that, you see here you have an example. The transfer RNAs, which are recognized, are differently approached. And you see, if you have a mirror here, the position of these transfer RNA here, like that. And so these are different sites. Here you have approaching by the minor groove, and this is a major groove. So all over the life of these two classes, you have two different mirror-like situations approaching. And so that was the structure. And these are all experimental results, of course. And so you see here an example of RNA. And so this is the key function separation, 2', 3', the chirality. And just to show you what that means approaching, these are some docking experiments which have been made with today's synthetase. Of course, they are not made to work together, of course. But just they show, you can see that here you have a class 2 synthetase, a class 1 synthetase. And you can put the tRNA, they almost can fit without excluding themselves. So in terms of evolution, it says, you know, that you had at the beginning, at least rather early, two possible ways to select, and then it's made. Then the second question, which was raised, I mean, at least here we have an answer, but then the other question is that here, if you look, you have the same number of synthetase in each class, 10 and 10. That was a rather surprising also observation, because wouldn't expect that. You know, why 10 and 10? I mean, it tells you something, but you wonder, you know, how that can be, because randomly, you know, you could have let's say 12 synthetase charging on 3' and 12. So definitely there is something behind that clean separation. But at this point, we have only model and speculation, because this is clearly a problem of evolution. So there are hypotheses which explain. I will give you two things. But before going a bit further, just to come about the problem which pertains to our observation in today's talk, that the fidelity of the reaction requires two recognition steps. The binding of the tRNA, of course, that's an important step, and the binding of the correct amino acid. These are the two conditions. And of course, these are, I mean, they could be linked in terms of cooperativity, allosteric control, but basically these are two different aspects. You could imagine that you charge the correct tRNA, you recognize correct tRNA, and you mischarge the amino acid. Then the tRNA will be mischarged. This happens. It's a mischarge, then you need some proofreading and correction experiment. So, but these steps are very important. For example, then for transfer RNA, it's rather easy now. We understand very well the specificity and the control. Here you have an example, which is the Aspartic System. It's a class 2 with 2 transfer RNAs. And you see that the recognition is achieved through 2 main regions of contact. That's near the active site, where you have the N, it's more common. And you have a module which recognizes the anticodon domain. And so here you have an intricate network of interactions which make very specific contact. And this is so sophisticated that it, most of the time acidities from one organism won't work in another organism. And this is very important for protection and things. So these is a key reaction, so you cannot have easily orthogonal systems. We'll see how we can make use of that in the next time. So the next step is the binding of the correct amino acid. That becomes a bit more tricky. Because here are the amino acids. 20, and take for example this one. It's a valine. You see the side chain here. And this one is a threonine. The difference between these two, sorry, I have yeah, here, sorry, that's a side chain. You see the difference between these two is just that one methyl group is replaced by an oxygen, hydroxyl. Sterically, they are almost the same. This is why, for these, if you have a pocket which can accommodate one of them, most likely the other one will bind also, even with less affinity, but you cannot afford to have mistakes. I mean, mistakes higher than 1 out of 10,000 would be very difficult to cope with. And this is an example, for example, of difficulty. You see this one, the serine, can also, the difference between this one and this one is just you miss one. So if the pocket fits this one, it will accommodate this one. So you can imagine, for example, that a pocket like the valine tRNA synthetase, which is built to accommodate the hydrophobic side chain, can easily exclude something with an hydroxyl. Well, easily, can. Easily. But the other way around, it's more difficult. In fact, these are the problems. That's why the enzyme have designed strategies for proofreading. And the most popular model is the double sieve model, where you have a first selection based on size here, and so the larger one are excluded, one which is larger, and the other, for example, here you have 2 that can bind. Then you have an editing site, which is designed for the wrong amino acid, the one you want to exclude. how does an enzyme work? Recognize the substrate, and if it holds the substrate long enough, reaction will occur. So the trick is to have a better binding site here for the wrong amino acid, then it stays longer and it is hydrolyzed, and then the tRNA is recycled. If it is the correct amino acid, it can still bind because but not long enough to be hydrolyzed. That's the basic idea about this control. And so these control mechanisms are with an added module, which is an enzyme, which has enzymatic activity, which does that. So again, when you look, since there are editing necessities in both classes, and you look at the reaction, we find again the same symmetry type. Here, if you have the catalytic domain on class 1 on the left side, you see, because in the minor groove approach, on the editing domain is on the other side. And here is the reverse. I don't know. No, sorry, I forgot. It's easy to understand when when you have the tRNA bound, you don't want the system to be released and search for another. That's a totally inefficient way. The easiest way is still bind the CCA, which is flexible, can move away once the reaction has occurred. Then it is released, the CCA end, and then it goes in the other active side. So it's a large cleft. It's a very, very efficient way. This side. This is why you keep the symmetry and orientation. So you just flip that and you go in. Here you flip and you go the other. And then check and release if it is okay. If not, it hydrolyzes, it releases, it can start again, recycle. Okay? So we have seen very sophisticated machines. Now let's go a bit further and see why the 20 and 20. So I will go very quickly because of the point. This is something rather bizarre observation and a rather interesting observation. Why do we have 10 and 10? The next question will be, why only 20? What happens with the others? So the first one, 10 and 10. Here you have the genetic code distributed with two types of analysis, because we are used to have genetic code and we look at genetic code with bases 1, 2, and then the distribution, and that's the third base. The color code is based on classes. You have class 1 and class 2. So, green and red. And so you do that, and if you have the classical thing, this is what it's a bit patchwork. You can redistribute the order. That's just a reorganization. And you will see that you have different clusters. You start to see that there must be some correlation in the order that the bases are selected for the genetic code. So here Marc Delarue, who was a key player in the finding of the sequence-based separation of classes and finding of pattern, he came out a few years ago with an hypothesis which was published in RNA, where here you have the principle of that. You take the first base, here can be any. Once you have the first base, you add the second one, and you begin to have some specificity. So if you say, now if I add my base on one side and on the other, I have two active sites here. Because here, right from the beginning, you have to accept I have no time to enter, that this is a very primordial thing. Right from the beginning, the primitive synthetase, which could have been an RNA actually at that point, because the reaction is very similar at that point. You could, the earliest primitive pseudo-ribosome would have been just a ribose. And you flip from 2' to 3' for elongation. So you need the two. Because once you have attached on one side, if you want to add, you have to go to the 3. So the partition is at the heart of the separation of the genetic code deciphering. So once you have one, you have one. So then if you want a connection with the bases, once you add a second base in terms of specificity, you have one, either 2' or 3'. You cannot have both at the same time. If you select one enzyme that does the reaction from the left on the other, then from the right you have another one. The idea is that each time you separate, you cannot change the order anymore, because if you add another one, you would have, if you want to change, you would have to go back. So the number of changes would be very, very important. So once you are trapped in an idea, it's unidirectional. And if you do that, you realize that you get, I mean, you can read the paper, a very important, a very nice explanation of how the partition occurs and how you have this separation. Now, there are surprises. You say, why 20? Why 20? And as it turned out, now we are today's present, not the evolutionary time. We have eukaryotes, archaea, we have for them this synthetase which is absent. This one is synthetase absent in archaea. So we have some missing things. I told you these were very important. Now you have organisms without it. So that's an important issue. I will not address that because, I mean, it has been addressed in several. There are explanations with other systems, but I will address the other one, which is a bit more of today's interest, at least. Why are we limited to these 20? Are there other synthetase charging different tRNA or amino acid? For the tRNA, the pool is there. The genetic code is there, messenger RNA is there. You cannot play so much with that unless you change the signification of the code. But in terms of amino acid, since you have the two separate things, you could imagine that you have another amino acid, another substrate, which is attached to a same transfer RNA. These are two different problems. And in terms of work, this approach, trying to change the specificity toward amino acid, has been an enterprise which started a long time ago. One of the pioneers in that field was Peter Schulz, who is now at Scripps, and I think it's Scripps, yes, Scripps in California, in San Diego, and he's a chemist, and he said, you know, if we modify the active site of the enzyme, then we should incorporate any amino acid, provided that we change. I mean, the only thing is that we have to do is to change the side chain, because the reaction only involves the peptidyl part. The side chain is the one which gives the specificity, but not the reaction. So that was the basic idea. So we should, but that turns out to be a quite difficult task, very difficult task. But they had some successes. And actually, if you look, there are some lots of paper, not only by Peter Wu, but theoreticians who calculate. You can go up. We ourselves in our lab try to switch from aspartyl tRNA synthetase to asparagine, which are very close. And if you look at the structure, they're very close. It seems very easy to understand. We went up to 6 mutations, very close. The first shell was completely mutated, and the second shell mutated, didn't work. So that shows you the complexity of things, the allosteric control. So it's not just the contact, you have other contacts. But as I said, this was really a large enterprise. Here you have a quotation from the paper, this paper 2003. Of course, this study started before that. At that time, here is an important step, is where the method became quite successful already. And there were some successes, and the idea here is to incorporate what is really the difficult step, the novel amino acid into eukaryotic cells. Why eukaryotic cells? Because if you can introduce specific amino acids with different functionalities, then you reach very interesting, you have in hand very interesting tools to study complex regulation systems. Just give you an example. You know, the control of transcription is achieved through a lot of modifications, either on DNA or on proteins. And one of the modifications important, for example, is acetylation or deacetylation. And you acetylate lysine of histone in the nucleosome core. This is how you trigger the reaction. So acetylation is one thing. If you could, it is very difficult to analyze these things, isn't it? But if you can introduce the acetylate residue at a specific location using this system, then you have in hand a very powerful tool. This is just an example. Of course, you can do more. You can do complex molecules. So here we have important things. So what you try here, you have an example. In this example from the same paper actually as previously, that's a stereo view, and is one of the favorite enzymes for doing that is tyrosine, tRNA synthetase. You remember I told you in the classification the tyrosine is the one which is a bit ambiguous about the selectivity, so it's the more tolerant about these 2' it's in terms of specificity. But that was not the only idea. If you look at the mutations, here are in the yellow part is the residues which were mutated. So you see this part where the reaction occurs at fix is maintained. So the enzyme can work and can bind the substrate even if you change the side chain, provided that you remove or change the residues which are there to ensure the specificity of the side chain. Of course, by analyzing the right pocket, you change the substrate here, and they did it, you know, and they incorporate these amino acids. You see, these are variation upon the tyrosine. You have always the same basic group, so you change these, and they could incorporate, and even that one modification. So you cannot do very sophisticated change, not any. For every change you have to be careful. This is properly designed. Now, told you, Jason Chin, who was in working with Peter, who will create his own lab, now is at MRC, and he made a very significant improvement to the method by being able to incorporate in yeast. Why yeast? Because it's a eukaryotic system. You can incorporate diverse natural amino acids using a tRNA synthetase, tRNA pair, which is rather specific. Now we come to what I said before. You know, in the normal, all the eukaryotes, ourselves, everything, we have 20 amino acids, 20 synthetase. That's it. But using bacteria and or archaean, this is a source of diversification. And there are bacteria which have these pair tRNA, is a specific tRNA, and of course this is a standard, I would say, with specific base for recognition, standard structure. This is a synthetase which is designed to recognize this amino acid, which is a non-conventional amino acid. So now the thing is to take that system and to bring in another cell to make the production. Because of course, if you use just these bacteria, you can only work with this, and you have no control at all. But if you introduce that in a eukaryotic cell, and you create a system, an orthogonal system, where you can make that work, control the work of this expression without disrupting the standard expression, then you have the tools. This is what they did. Okay. So here's a principle. That's a reference, a last year reference. And so you see, these are the amino acids that they try to incorporate. I must say, to give the credit, the first who found the first group who found this pair of pyrolysyl tRNA acidities was the group of Yokohama in Japan. But the phenotype was a bit weak. So Jason Chin used that, but improved, and this is most efficiently implemented with a lot of things that I will show you, which makes it a very efficient tool. And so this time we have, with this enzyme, so the basic chain, it's a lysine with modifications here. For example, this is the most. So the backbone is the lysine. So we have a system which can incorporate lysine, modified lysine, which is exactly, I mean, the deacetylate lysine or the modified lysine are very useful to control transcription, for example. So this is exactly the tool we need. One, but in principle, we can do a lot more. So you need to first adapt the transfer RNA, then to bring the proper synthetase and make it work in an orthogonal way. So first you have to create a functional tRNA CU in yeast, because you want to transfer. This transfer RNA was working in archaeon. I forgot the name of this bacteria. Okay, sorry. It is in the slide, the name of the bacteria, it's in the paper. But you have to transfer the transfer RNA. The transfer RNA does not exist in yeast. So this is the construction. Consensus, and this is the modify box of bacteria. I keep forgetting the name of the organism. And these are several constructs for the expression construct that have been made, and these are the control. And you see here you have northern blot to see the expression from the virus construct, and you see that this one is working more efficiently, and then you have the fifth one which is giving some, and the other don't work. So there is one construct which works, and very important progress. So you have the tRNA. tRNA as a partner. So this time we have a specific partner. You remember what I told you? Transfer RNA recognition is an important step. This one is specific for its partner synthetase, but it's not recognized by the synthetase of yeast, not as efficiently. You see, that's important. So the recognition, and then you incorporate. That's why it's orthogonal yeast, and not, but this one is not. So direct incorporation of the alanine system. So we have here the working that shows where you have the amino acid. This is the working system with the construct, and these are the two constructs which work. Control. Now that you have the synthetase, the tRNA, you can run, and if you want an efficient system, the next step is to have a network of regulation. The same group. I recommend you to read these papers. You know, a transcription translation network. So, parallel and independent system for synthetic biology. We are talking of synthetic biology, but this is biotechnology. So create, and the principle there can be applied to a lot of other things. You can think of a very completely diverse polymer if you wish to, if you need. But the first application is the one I told you, which is an obvious one, is to have a tool, an efficient tool to analyze the modification, the post-translational modification on proteins. So you need an orthogonal. For that, you need parallel independent system. So they combine the orthogonal transcription efficiency of T7 polymerase and translation by orthogonal ribosomes. Of course, if you want to have an independent system, I told you tRNA, CDTase, ribosome. And then you have the complete, if you embedded that in the cell, you have something, and if you can control. Here is the network, that's the orthogonal, that's the normal, and the orthogonal gene expression is insulated from cellular gene expression, of course. You don't want them to mix. You have specific action, you want to trigger that when you wish and to prefer. So hand function here. Then the construction of an orthogonal hand function to adding what becomes here. In the details you see, this is the end to include, to make it work through the production. Here these are the control, the sequence selected have 3 rounds for sorting the ribosomes. And then here you have the different transcription, shows the logic and also the results. So this is continuation of orthogonal transcription. So this is the end system now of the orthogonal transcription translation. So now what is very important in transcription regulation is also to have a feed-forward loop. I have no time to enter these things, but you know, in terms of regulation, where you have the circadian clock, for example, has been studied, neural network, you have these feed-forward loops that are important for the control. You want a system which controls, so you include that. And this is what you insert, and the genetic circuit that are made, they made that. So it's a fantastic. They want to design, they use a ribosome, a specific ribosome, and so the ribosome RNA affinity tag, just this is to purify the right ribosome, because they want to include. You see they put a linker here on the right. The tag is here, so that you don't touch the structure, you just pull it out and purify. And the orthogonal ribosome affinity purification strategy, you bind to the tagged ribosome and you extract. So this is just, it's all classical canonical step. And of course, now you see the system is working. It works with in vitro termination assays to check that the system is really improving, decrease affinity for RF1. If you read the details I have in this paper, you will see the whole explanation. So they implemented the system, and the system is working efficiently to include, and this is a major achievement. Very important, because that you can do that for other systems. So the idea now is to use the power of the selection of nature with bacteria to select the bacterial that generate, yes, I'm trying to feel, the correct CDTases that work for exotic substrates. And there are now pools available. This is the studies which are going on more and more, and these bacteria are very efficient to make evolution work efficiently for specific purposes. Of course, these would be very harmful for us if they would do that. But the cycle of the evolution is very quick for these bacteria, so you can generate. So you don't have to find, like what I showed you at the beginning, smart chemistry to say, okay, if I mutate that, I mean, it's still available, but very complex. Nature does that much more efficiently for you. Once you have that, the trick is to import it there. And this is a strategy which was designed, by no means limited. It's a first step. I'm sure we can do more, and you can think of lots of applications. So it is really expanding the genetic code, but not to have a 22 amino acid genetic code for us Euronis. is to make use of the genetic code for specific additional and exotic or even artificial amino acids that can be then used to incorporate in protein at a given place. For the isolated tRNA, you have to change the to adapt the messenger RNA, of course, also. And you can make artificial messenger RNA with specific target points. So there is an extremely large pool of application that you can imagine now in biotechnology and in research. And I think I stop there. Thank you.